We've all been in a situation where we felt anxious, or exhausted, or just stressed out in general. Whether it be anxiety over that meeting you're losing sleep over, or exhaustion from studying for that exam all night, or just feeling stressed because you had to prepare for a 7-minute presentation about your research. Well, we've all been there. And in these times of extreme stress, when you thought nothing else can possibly go wrong, you end up with this, an annoying little cold sore. I'm Susan Park, and I study the virus that causes these cold sores. And today, I would like to talk to you about how this virus can actually cause a very serious debilitating eye disease called herpes stromal keratitis. Herpes stromal keratitis, you're probably not familiar with this, so let me explain. I want to take you back to your grade school health class. You might be familiar with these slogans, warning you about the dangers of sexually transmitted diseases. And what you might remember from this class is an example of a notorious pathogen known for causing oral and genital sores called herpes simplex virus 1, or HSV1. HSV1 is most commonly spread through oral-to-oral contact, and 70% of adults are currently infected. That's almost 4 billion people walking around infected with this virus. And while not all individuals who are infected have symptoms, many of them present with mild symptoms like this, called cold sores. But what they do not teach you in your grade school health class is that this virus can actually cause very serious diseases. For example, if the virus can infect the brain, it can cause a disease called encephalitis, which can be fatal, especially in newborns. When the virus infects the surface of the eye, it can cause severe vision impairment or blindness, which is called herpes stromal keratitis, which is a focus of my research. Herpes stromal keratitis, or HSK, is a leading cause of infectious blindness in the United States. There's approximately 40,000 people newly affected each year, and yet there's no targeted treatment available. The current treatment regimen consists of general oral antivirals, the same ones to treat mild cold sores. So we're in a dire need to come up with a new therapeutic for this disease. HSK is caused when the virus infects the surface of the eye called the cornea. And the cornea normally functions to refract light, so it needs to remain transparent to do its job. However, what happens in HSK patients is that as the disease progresses, and there's more and more damage to the corneal tissue, the cornea gets less and less transparent, causing vision impairment and complete blindness. So how does this damage to the corneal tissue actually occur? Well, as you can see here, in a normal healthy cornea, there's a limited number of immune cells present. But when the virus infects this tissue, the body wants to fight off the invading pathogen, so it employs a large number of immune cells to the site of infection. And while these immune cells are successful in clearing the virus, it persists and causes chronic inflammation and the bulk of the symptoms as seen in HSK. So HSK is described as an immune-mediated disease because it's not the virus, but the immune cells that persist in the eye that cause chronic inflammation and the symptoms of this disease. So knowing this information, my lab asked the question, then what drives this chronic inflammation? If we can find this out, then we can block it or reverse it to alleviate the symptoms of HSK. So to start to answer this question, we focused on the immune cells that enter the eye during the disease. As you can see here, these are 3 different types of immune cells. And what you see in these colorful rectangles are the proteins that are expressed on the surface of these cells. And these proteins tell the cell what to do and where to go. So finding out the functions and dysfunctions of these proteins will allow us to determine why these cells get to the cornea and persist, causing chronic inflammation. And in order to do this in the lab, we use a mouse model of HSK, which actually largely mimics the symptoms in humans. We infect the mouse in the eyes with the virus, and then perform various experiments to determine the identity of these cells, as well as the magnitude they persist during disease. So from these experiments, we identified a protein, a specific protein that is involved in driving this chronic inflammation. And how we did this is when we infect a mouse, a normal control mouse, as you can see here, that can express this protein, we observe increased inflammation and subsequent increased corneal damage. However, when we infect a mutant mouse that can no longer produce this protein, interestingly, we saw decreased inflammation and largely diminished corneal damage. And what this tells us is that the presence of this protein causes this chronic inflammation in HSK. We are currently working on looking at the molecular mechanisms of this protein with a broader goal of identifying this as a potential therapeutic target. And although our lab is in its early stages of this research, we are hopeful to be contributing to developing a novel therapeutic for HSK treatment. Thank you.